Hi, this is Dr. Sheldon Jordan, and I'm here to talk about a project which I'm very passionate about, and that's finding ways of delivering biologics to the brain in ways to try to optimize brain function. Here are my affiliations and conflicts of interest. And here's my disclaimer. Various techniques have been used to try to deliver stem cells and exosomes to the brain, including intravenous techniques with mannitol, nasal applications, lumbar puncture, direct injection into the brain parenchyma, injection into the ventricular system, cisternal puncture, arterial and venous catheterization techniques, and lastly, focused energy, including repetitive TMS, transcranial magnetic stimulation, laser light therapy, and transcranial ultrasound. Getting biologics into the brain has always been quite a challenge because of the blood-brain barrier. In other words, when something is given systemically, there is a strong tendency of the brain to prevent things from penetrating and producing its potential therapeutic effect. So we're not just simply interested in getting biologics across the blood-brain barrier. We actually want to get them to specific places within the brain. We know that stem cells exist lining the ventricles and also adjacent to the hippocampus, and these stem cells may be very important for our ability to remain youthful and to learn new things as we become older. Research over the years seems to show that the ability to maintain the stem cell niche around the hippocampus and around the ventricle appears to be absolutely critical in maintaining cognition as people age. So when there's loss of these stem cells adjacent to the hippocampus and adjacent to the ventricles, there's a progressive deterioration in cognition that will progress from normal aging to mild cognitive impairment and lastly to Alzheimer's disease, as is suggested by this diagram. So the big question is, how do we preserve these stem cells and maintain their ability to proliferate and support a functioning brain, and to maintain cognition as people become older? Well, there are certain lessons that we learned from the parabiosis experiments many years ago. The parabiosis experiment acts something like this. You can take two mice and you can connect them together, including their bodies and their circulation. Now, when you do that and you take a young mouse and connect it to another young mouse, nothing very interesting happens. They both stay young. Likewise, one can take two old mice and connect them, including their bodies and their circulation, and then you have two old mice who both stay old. Now, something very interesting happens here. When you repeat this parabiosis experiment and you connect an old mouse with a young mouse, something very interesting happens over the course of some time frame. The old mouse begins to act younger and look younger, and the young mouse starts to look older and act older. When it comes to understanding how parabiosis works at the brain level, the group headed by Dr. Villeda and his colleagues was particularly interesting in determining what happens to the part of the brain, the hippocampus, that's responsible for memory and loss of memory as individuals become older. So let's see what happens inside the brain with this parabiosis experiment. We're now looking at histological slices taken from the part of the brain that's relevant for memory, the hippocampus, and we're staining for the stem cells. Now, in this subgranular zone in the memory center of the brain, we can see many stem cells in young individuals. So if you look at that left upper box, here we have two young mice that have been hooked together with this parabiosis experiment. And since they're both staying young, if you take a histological slice of their hippocampus, we see a lot of these dark staining stem cells. On the other hand, if you look at the lower left box, here we see the result of two old mice that have been hooked together. In this case, there's only a couple of remaining stem cells. In other words, as a mouse becomes older, they lose almost all of their stem cells, and this is also true of humans. And not unexpected, these older mice have a hard time learning new mazes. They're not as active in their family groups. They're not able to be cognitively intact as they become older. Now, here's an interesting thing. If you look at the young mouse who's been hooked to an old mouse, and you look at the bottom right slice of the hippocampus, this is actually the slice taken from an older mouse, looking at his hippocampus. And what you see, there are many more of these dark staining stem cells than you would have seen in an old mouse by itself or an old mouse hooked to another old mouse. So in other words, when you hook up a young mouse to an old mouse, they start to grow new stem cells. And that's very interesting because that allows the mouse to learn new mazes and to be cognitively younger acting. Now, when you've done the parabiosis experiment, those animals have been hooked together for quite a long time frame. So there's plenty of opportunity for the active factors to transfer from a young mouse to the older mouse. So in other words, there's a constant bathing of the older blood with younger blood. So what happens if I were to take some potentially regenerative factors such as stem cells and inject them into a vein? For example, we could take some stem cells derived from a young individual and simply put it into a vein and see what happens. So here's a study done in a dog by Dr. Kreichman and his colleagues in 2005. And you see what happens on day 1, day 2, and day 7. In this case, the researchers have purposely caused an injury to the heart. And the heart is sort of in the middle of the chest there. The color that you see is showing exactly where the stem cells hang up after intravenous injection. So in the very first day, we see a lot of colors in the lung. So as the stem cells are injected intravenously, many of them hold up in the lung for the first day or so. By day 2, we see a lot of color in the spleen and liver. In other words, the stem cells, to some extent, are able to wiggle out of the lung, and they end up getting taken up by the spleen and liver. And it isn't until day 7 that actually you see a little color in that area of injury that they caused in the heart. So after a very long time frame, a very small amount of stem cells will eventually find their way to a site of inflammation. But the amount of coloration there is in some way directed to the amount of stem cells that actually end up there. And there's actually a very low dose that actually ends up there, because much of the stem cells have been taken up and disposed of by other areas in the body, including the liver and spleen. So there's very little left over to actually find its way eventually to an area of inflammation in the heart in this case. Now, I'm sure that many of you are very familiar with exosomes. So exosomes are little particles that include a variety of regenerative factors, mostly non-coding RNA that is encased in little lipid membranes that can circulate around the body. They're given off by stem cells and other cells in the body. So if you were to take some exosomes that have been derived from mesenchymal stem cells, we can then inject them either directly to an area of inflammation, or we can give them intravenously. In this first experiment, if we inject it into an area of injury in this particular mouse, one can see that the exosomes pretty much stay in place. So if there's an inflammatory signal and one injects directly into a spot, they'll stay there. So we have the first hour, 12 hours, and 24 hours. There seems to be pretty much a constant remainder of the exosomes which are tagged, staying in an area of inflammation where they were directly injected. By contrast, if you give these same exosomes intravenously, most of them will go to the liver and spleen. Now, exosomes are quite smaller than stem cells, so they pretty much pass right through the lung in this particular rodent model, and they end up in the spleen and liver. And even by 24 hours later, there's very little that ends up going to that area of inflammation in the right thigh muscle. So in other words, the yield of exosomes that actually end up in our target zone appears to be very low when given intravenously. So this brings us to our next idea. If we can't get things into the brain very easily by giving it intravenously, in other words, a lot of it gets taken up by the lung the first day and by the spleen and liver for subsequent days, leaving very little else to get to the brain. How about if we try to do something much more proximate to the brain, such as having a nasal insufflation of biologics to see if they'll actually get things directly to the brain? So it looks like the top of the nasal cavity has only a thin connection with the rest of the brain, and perhaps biologics can be induced to travel right through this thin bone which has fenestrations in it called the cribriform plate. And it's also been proposed that perhaps some of the material can go directly through the fenestrations in the cribriform plate, but there may also be a pathway adjacent to blood vessels and adjacent to nerves, and that may be a way of entry into the brain. Well, not so fast. It turns out that those fenestrations in the cribriform plate are huge in mice and rats, where some of these experiments have been done. But when it comes to humans, and particularly older humans, these fenestrations in the cribriform plate tend to calcify and get clogged up. So there isn't that entrance into the spinal fluid space as one would like to see. So here we can look at the cribriform plate in a relatively young human, and on the left side, one can see enormous fenestrations where you might think it might be pretty easy to get things to go through and get into the brain. On the other hand, if we look at the right side of the illustration, we see the cribriform plate from an older individual, and these fenestrations either occlude completely, they become calcified and ossified, or they become extremely small. So it's very difficult to expect that biologics can actually find their way from the nasal cavity into the brain. So it gets even more complicated. So let's say, even in an ideal situation like a young rodent, where these pathways might be patent, it turns out that when there is a nasal delivery of biologics, they don't simply distribute to the brain in areas that you might be interested. They go to specific places. So if you look at this early article that was published by Doofus, 2003, he had the interesting experiment of looking to see exactly where these biologics end up when they're insufflated through the nose. And it turns out they distribute in only particular places and they're not seen in other parts of the brain. So there isn't an even distribution, and certainly not getting to the stem cells lining the ventricles or adjacent to the subgranular zone in the hippocampus like we would like to see. So if you try to correlate the distribution pattern of what was found in the Doofus experiment, the delivery appears to go along vascular routes. So in other words, either perivenous or periarterial, but if you look at a venogram or an arteriogram in a rodent, it appears that the major distribution goes along the vascular pathways and doesn't necessarily get to some of these key areas like the stem cell containing areas that we're most interested in treating and targeting. The group at Stanford did an experiment in humans where they directly injected stem cells for the treatment of stroke. And they appeared to show some success with direct installation of stem cells into parts of the brain adjacent to an area of injury. What's particularly interesting about direct injection of stem cells into the brain, they actually don't take over the role of damaged cells. Rather, what they do is they seem to give off growth factors and exosomes, and this appears to stimulate local cells to proliferate in some fashion and to help the healing process along without a direct replacement of the damaged cells with the newly introduced stem cells. Here's a histological study done by Jerome, which showed that the stem cells appear to die after some time frame, and rather than replacing existing cells, they seem to produce a proliferation of cells in the area, including resident stem cells in the subventricular zone, as well as a proliferation of astrocytes. Here's a direct injection technique, which I'm particularly fond of, that was performed by the Danjun Kai group at Albert Einstein. There was a direct injection of exosomes into the region of the third ventricle in mice. Here's the experiment by Dr. Kai. If one takes a needle, we can provide an immunological agent into the third ventricle, and one can destroy the stem cells adjacent to the hypothalamus that are responsible for maintaining a youthful signaling within the hypothalamus and maintaining a youthful age. Once these are destroyed, the mouse becomes old. Their hair falls out, they become sexually inactive, and they become cognitively impaired. They can't learn new mazes. But here's the really interesting thing about this experiment. One can take this mouse, where the stem cells have been destroyed around the hypothalamus, and one can apply exosomes from a young mouse. These exosomes are enough to reprogram the hypothalamus so the mouse remains young. Their hair grows in, they become sexually active, they become cognitively performing well, learning new mazes. Injecting things directly into the brain seems pretty invasive, and it doesn't seem to be the kind of thing that really could be applied to a large population. On the other hand, alternative techniques have been used over the years. Here's a technique that I've used over the years, including a cisternal injection. So here one can get access to the spinal fluid space at the base of the skull and inject stem cells or exosomes. Here's one at the C0, C1 level. And here's an approach between C1 and C2. When people, including myself, have used these techniques, the results have been quite variable. And I think one of the major issues is that the major vector of spinal fluid flow is actually not into the brain from the skull base, but rather away from the brain, percolating around the outside and ending up being absorbed around the surface of the dorsal aspect of the cortex. So that this does not appear to be a good pathway to those stem cells that line the ventricular system or are adjacent to the hippocampus. Rather, the bulk of fluid appears to be going in a downstream fashion that would take exosomes and stem cells going in the wrong direction, away from the ventricular lining cells that we would like to otherwise target. One other option was to place an Ommaya shunt directly into the ventricle, and this has the advantage of allowing the bulk spinal fluid flow to move the injectates along with spinal fluid and bathing the entire area that are lined with stem cells, and even bring things potentially towards the hippocampus and subgranular zone, where some of those important stem cells reside, as well as potentially into the stem cells lining the hypothalamus and elsewhere. This approach has been used effectively by Dr. Duma and his group, where he placed Ommaya shunts in individuals with Alzheimer's disease, and he was able to show some effects in terms of maintaining cognition in some of these individuals that were expected to have worsening over the course of years, but rather had stabilization or even some improvement. For a moment, I wanted to circle back with this idea of epineural application of at least exosomes. It appears that exosomes can be taken up by the nerves and carried back towards the nuclei and into the spinal cord and or brain, where additional therapeutic benefits might potentially occur. Here's an article by Wren and his group that demonstrated that there can be an axonal uptake of exosomes and there is actually the presence of retrograde flow back into the nucleus within the spinal cord, so that some of the elements that are carried by the exosomes can be tagged for, demonstrating a selective delivery to the central nervous system from a peripheral nerve injection. Here we're showing what an epineural injection would look like. Here I'm taking a needle, and you can see me inserting it in the center of the slide, very close to the subclavian artery in the center, and the neurovascular bundle was just off the left of center. I will inject a little bit of saline so that we can establish a plane of dissection, and you can start to see the neurological bundle of the brachial plexus come into view. We can then take our needle tip and actually get within the neurovascular bundle and inject adjacent to the epineurium, as we're seeing right at the moment now, where you can get right next to where the nerve is traveling. And in that type of a dissection, one can start to consider putting in various kinds of biologics, which has the potential of being carried back to either the spinal cord or in the place of, let's say, the vagus nerve, the facial nerve, or trigeminal nerve. One has the capacity of bringing back some of these biologics to the nucleus in the brain. Arterial injection has also been thought about as a potential way of getting biologics into the brain. And there are several studies that have looked at using the arterial route as a way of delivering biologics to areas of brain ischemia and brain injury. Initially, there was some concern that this would cause some problem with blood flow and potentially produce strokes. However, more recent investigators have found that it is possible to use this type of deployment in a way that appears to be safer and potentially effective. There's another technique that everyone's sort of forgotten about. This is a technique that I worked out with John Frazee and some of my colleagues at UCLA many years ago, is using the venous route to get things into the brain. In this technique called venous retroperfusion, one can place catheters retrograde into the jugular vein with a occluding balloon, and one can actually produce reverse filling of the venous system into the brain and down to the capillary level. We've done this in baboons, and we also did this in humans. This was published in the Stroke Journal many years ago. The advantage to this in the setting of, let's say, a stroke, it turns out that capillary pressures are very low downstream from an occlusion on the arterial side, so that when one delivers things via venous route, there's actually selective blood flow to the area of injury, which seems to be an absolutely perfect way of thinking about applying biologics for the purposes of regenerating damaged tissue in a setting of stroke. Now, I'm particularly interested in relatively non-invasive, relatively inexpensive, accessible, and safe ways of providing biologics to the brain and facilitating efficient delivery. So in this case, I'm interested in various kinds of focused energy sources to help with delivery. Focused ultrasound is a technique that we've been working on for many years. This is one of our first papers that was published in 2019 in the Alzheimer's and Dementia Journal. And here we're using focused ultrasound to the hippocampus, among other structures, to help facilitate the delivery of small molecules to the brain in people with Alzheimer's disease. The idea was to take a tyrosine kinase inhibitor, which was very poorly penetrating into the brain, and facilitate that delivery using the focused energy affordable with ultrasound that could be applied in a routine clinical setting. So the experiment in the animal labs we work with, such as with Karen and Booty at the City of Hope, we would get a rat in a stereotactic frame, and then the ultrasound device is placed stereotactically in the right position, in this case over the hippocampus, and then we provide the ultrasound energy to our target zone, followed by an intravenous injection of an injectate that has exosomes in it. We'll look at some of the results of this experiment somewhat later. It can be done in a routine clinical environment with humans. So here we're showing you what a head holder would look like when applied to a transtemporal window, which would hold our ultrasound probe. The setup with a human would use a technology called optical tracking. You can see in the corner of the room, the far left corner of the room, that there is a virtual brain that has the data from an MRI scan that was taken, of course, offline in an MRI scanner. That data set is ported to this optical tracking device, which uses an infrared camera to register the real head in space with the virtual head, so that wherever the technician is pointing that pointer on the real head, one can see where the targeting is in the optical tracking device in the virtual head. This is the same kind of technology that's used by neurosurgeons to determine a pathway to resect a tumor, for example. So we can use the MRI scan data taken before the clinical visit and use that data set to actually target our ultrasound device, just like a neurosurgical instrument, with millimeter type accuracy. In other words, we don't have to do the targeting inside an MRI scanner. The MRI scan is done beforehand, and the data set is used in a routine clinical environment so that we can accurately target any particular nucleus in the brain that we might be interested in. So here we're looking at an arterial spin labeling, and if you look at the brain as a baseline on the left side, the arrowhead is going to be our target zone on one side for the hippocampus. In this case, we actually ended up targeting the hippocampus on both sides. And once one goes to the right side, after a certain number of minutes of ultrasound energy is applied to the hippocampus, the arterial spin labeling demonstrates a marked increase in blood flow only in the target zone, as demonstrated by increased yellow coloration, which is related to a quantifiable perfusion value. So in other words, ultrasound to the target zone increases blood flow, and when we do this quantitatively, we have been appreciating that the blood flow can be increased as much as 8 times the baseline value. So if something is, for example, starting out with 20 or 30 cc's per minute per 100 grams of tissue, one might see values well over 100 cc's per 100 grams of tissue after treatment with ultrasound. The nice thing about using this kind of low intensity ultrasound, it can be done without producing cavitation or ablation. In other words, this is not high intensity focused ultrasound that's designed to produce a lesion, but rather this is diagnostic level ultrasound. So we would be looking at something like 720 milliwatts per centimeter squared, which is diagnostic value ultrasound, very safe. 40 plus years of experience using this level of ultrasound is sufficient without using microbubbles or any other technique. Simply using diagnostic ultrasound for a long enough time frame of, let's say, an hour at 2 megahertz, one can get delivery of exosomes and stem cells to the brain. Focused ultrasound appears to stimulate the formation and release of various compounds in brain endothelial cells. In fact, it's been shown by several investigators that ultrasound actually increases the adhesiveness of cellular elements to the endothelial luminal wall. So think of this, with ultrasound, when a cellular element rolls by an endothelial lining, it'll stick to the surface of the endothelium. This is followed by an invagination and a transcellular migration of the cellular element across the endothelium to the brain side of the vascular lining into the brain parenchyma. Now it's not just simply increasing of perfusion in the target zone that is created by the ultrasound energy. And before going further, I would point out that in humans, when we've looked at DCE imaging of permeability, there really isn't very much change in the amount of leakiness of the blood-brain barrier. So we're not simply tearing apart the tight junctions. And also, I would point out that because we're not tearing out the tight junctions, there's no bleeding and no edema. So, in concert with the City of Hope group, including Karen and Booty, we did the following experiment. We looked at a different duration of ultrasound stimulation to a rat brain hippocampus. And we performed an experiment with fluorescein-labeled dextran. And what you can see with 20 minutes of ultrasound in the left columns, there's actually very little fluorescein label uptake. But when you get to 40 minutes and finally up to 60 minutes of ultrasound at 2 megahertz, one can see this clumping, bumpy adhesion of dextran labeled with fluorescein to the endothelium. So in other words, it appears as if the ultrasound actually increases the adhesiveness of the endothelial lining without tearing open the tight junctions, without causing bleeding or edema. And here's the most important slide. If we look on the left histological slice of a rat brain that's not in the target zone of our ultrasound, so in other words, this is the control side of the brain which does not get any ultrasound, one can see a few bright spots in the histological field. And those tiny little bright spots are the very few exosomes that get into the brain just because they're given intravenously. So there's perhaps up to a 5% penetration of exosomes into the brain without any particular technique used to facilitate their entry. But look at the amazing contrast there is with the histological slice on the right. Here we see something that looks like a starry sky. It looks almost like a galaxy. These are all exosomes that have been labeled in the target zone. So this is the side of the brain that's getting the focused ultrasound at 2 megahertz for 1 hour, and one sees an enormous increase in the delivery of exosomes to the target zone after they had been given intravenously. So in other words, the focused ultrasound is done first for 1 hour, and then there's an injection into the tail vein of the exosomes, and when we label them, we say an enormous facilitated delivery of exosomes only to the target zone and not to the control side. So photomodulation is something that has been considered for this purpose. Laser light can be absorbed by brain tissue, and there can be substantial penetration depending on the waveform that is selected. So if one uses a wavelength of 1064, one can get penetration with devices that are on the market that go as deep as 7 centimeters from the brain surface. Many people have already looked at slightly shorter wavelengths at 900 or so nanometers. Transcranial magnetic stimulation can also result in temporary increase in blood flow that might potentially facilitate the delivery of stem cells and exosomes. However, it's not clear if magnetic energy increases the adhesiveness of the endothelium in such a way that stem cells and exosomes will stick to the target zone and then penetrate by transcytosis into brain tissue. There does appear to be some evidence that photostimulation and, as we discussed, ultrasound energy can increase adhesiveness and facilitate transcellular migration of biologics. However, it's not clear if this happens with magnetic therapy. So let me summarize what we've discussed. Preclinical experience suggests that stem cell niches in the brain may be rejuvenated, and cognitive performance may be enhanced with recurring exposure to humoral elements that may include exosomes and other growth factors transferred from youthful sources. This is what we learned from the parabiosis experiments. Direct injection of signaling cells into the brain appear to release exosomes and other factors in restoring dysfunction to brain, but they don't typically survive nor integrate into functional pathways as replacement neurons. Restoration of brain function requires targeted therapy, as sites of failure will depend on clinical circumstances. In other words, localization matters. Just simply getting material across the blood-brain barrier or into the spinal fluid space isn't enough. We need to get our biologics to specific areas, such as those parts of the ventricular lining or the subgranular zone in the hippocampus or the third ventricle lining area where the stem cells reside, where rejuvenation has to take place in a way that can restore brain function. Simply getting material across the blood-brain barrier or into spinal fluid doesn't necessarily get it to the area of interest. Inexpensive and accessible, safe, and reproducible targeted delivery of regenerative materials may be accomplished in humans with a deployment of focused energy sources such as ultrasound and light. This is an area of great interest, and we look forward to see how this field develops over the coming years. Thank you so much for your attention. It was great to discuss this with you. I look forward to working with you on a closer level over the coming years.